everyone. We're going to get started. I have the pleasure to introduce Professor Dave Fredriksson, who joined our School of Marine Science and Ocean Engineering and the Faculty of Mechanical Engineering here at UNH back in August 2022. He's also the director of the Center for Sustainable Seafood Systems. Dr. Fredriksson brings over 2 decades of experience developing ocean engineering methodologies for the farming of finfish, shellfish, and macroalgae with the goal of supporting seafood security for both the United States and abroad. His underlying passion is to grow new sustainable production methods and educational programs that will enhance both local maritime communities and the ocean environment. Professor Fredriksson has been the principal investigator for numerous research efforts with projects most recently funded by the Department of Energy, World Wildlife Fund, National Oceanic and Atmospheric Administration, and the Atlantic States Marine Fisheries Commission. So please give a welcome to Dr. David Fredriksson.
Friday afternoon. All right, should I turn up the volume a little bit? Okay. All right, so thank you so much. It's an honor to be here with you all today. My name is Dave Fredriksson. I'm a professor here at UNH. I had a pretty long career between my stints at UNH. I was at the Naval Academy for 17 years, but before that I got my PhD here, I got my master's here, so I'm very familiar with the landscape here in Germany. Right, Igor? Yeah. Thank you for the introduction, so I won't repeat the introduction. So I'm going to talk today about sustainable seafood approaches for the extensive coastline of New Hampshire. Yes, we do a lot here. We do a lot here in New Hampshire, don't we? It's a beautiful place. And actually, I don't do any of this work, and so I can't claim to say that any of the stuff that I'm showing in this presentation today, I did, because I pretty much didn't do any of it. But I do have a pretty extensive team that I work with of scientists and engineers who are really a fabulous group of people to work with. They're hardworking and they really know how to get the job done. And it's actually an absolute pleasure to work with all of these folks and more. Okay. Oh boy, I'm getting touchy. There we go. Before I start, however, I'd like to make a statement of ethics here. And I do this before all of my presentations. Our team that we have offshore in our coastal waters, we're constantly working with animals, specifically vertebrates. And so we are passionate about the health and welfare of these animals. They do have feelings and nervous systems. Even though we harvest them for sustainable food purposes, I want to let everybody know that we have oversight by the Institutional Animal Care and Use Committee, IACUC, and all of our work that we do is licensed by the New Hampshire Fish and Game. And this is our IACUC protocol number if you're interested. So now for the 3-hour tour. If you are old enough, you know what I'm referring to. That's a Gilligan's Island reference. Yes, I got a couple thumbs up there. Thank you so much. All right, we know what the age demographics are here, right? All right, so here's the 3-hour tour. UNH, our center, has 4 permitted aquaculture sites. And I'm going to go through what we're doing at all 4 of these sites. I'm going to have a lot of focus on number 1, which is our integrated multi-trophic aquaculture system. Site is about 3/4 of a mile around the point of Newcastle where the Coast Guard station is. We also have an education site with the Seacoast Science Center. One of our sites is in Maine waters. This is a Maine limited purpose aquaculture site. We are doing some stuff in Maine. So we're expanding. Are we going to take over Maine? Probably not. But we want our toes in their water, though. Absolutely. And we are really excited about our offshore site. This is in 35 meters of water, and it's permitted for 130 acres. It's a really unique situation for us to do our work. I'm going to do a lot of focus here on site number 1. And so to get to site number 1 and all the other sites, we start at Newcastle at the Judd Gregg Marine Research Complex. And we know it's underneath the umbrella of the School of Marine Science and Ocean Engineering. And our vessels are located there. You can see the Gulf Challenger. The Hassler is also home ported there. That's a NOAA research vessel. And this is our vessel right here. It's the RV Tigo. It's an amazing boat. It's 33 feet in length. And it screams. It's got twin 250 outboards on it. We can get to the Isles of Shoals in about 15 minutes in flat water. It screams. And it's an amazing vessel. So from this site, and typically using this vessel, we make our way around the Marine Research Complex. Most of you know where that is, and this is the site of our integrated multi-trophic aquaculture program. So, integrated multi-trophic aquaculture. Now, I'm an engineer, I'm not a biologist, so I'm not going to define all these terms. All these terms are new to me, so you're just going to bear with me. We do have some biologists in the audience, so you can ask them. But this is the concept. The concept of multi-trophic aquaculture is to grow a high-valued fed species. By growing the high-value fed species, it's being fed. So you're introducing stuff into that system. That stuff comes out of the fish as nutrients or wastes. But what we try to do is to offset those nutrients, the waste, by using extractive non-fed species. And so at this site, we're permitted to do all these different things. So we're permitted to do steelhead trout. That's our high-value species. We're also permitted to do blue mussels, oysters, and scallops. These are shellfish. They're non-fed. They extract nutrients from the water. And we're also permitted to do sugar kelp, dulse, nori, and sea lettuce. These are brown, red, and green types of macroalgae. And we also have lumpfish here. Michael Doherty over here is an expert in lumpfish. And we have a permit to put lumpfish into our system because what they do is that they're feeder fish and they actually can pluck off parasites like sea lice off of our product fish. And I can't forget, this is an echinoderm, a sea urchin, right? Okay, I just learned what that was. That term, I love saying echinoderm. We're permitted to do all these things at this research site. The system is a very, very small system that we have deployed at that site. It's about 6 meters wide and about 4.5, 5 meters deep, and there's 2 bays. We put about 4,000 steelhead trout, 2,000 in one and 2,000 in the other. We also can hang 72 meters of mussel droppers off the system and kelp lines as well. The mussels and the kelp are there for extractive purposes, though it's really not enough to assimilate all the nutrients coming from the fish, but I'll talk about that later. But this is the concept. I'm going to now focus on the steelhead trout part of it. And so our steelhead trout that we have in the system, they're actually in the system right now. We've been harvesting all summer long. I'll show you some pictures to that. But we get our strain, our eggs from Washington State. This is the river and strain. These are the trout eggs. They are rainbow trout when they go into fresh water and they get hatched and raised in a nursery in Ossipee, New Hampshire. And when they're about 300 grams or so, we transition them from fresh water into seawater. And I believe that's called the smoltification process, where they start changing their body physiology to handle the salt water as opposed to the fresh water. And they grow quickly in seawater. So a 300-gram fish is about that big, about 0.6 pounds or so. And when we do the fish transfer from Ossipee to New Hampshire, we put them in these exactics on the back of a UNH truck, and we transport the fish. We actually have to pump pure oxygen in order to maintain the oxygen levels in these exactics. We load it on fishing boats. We really use the local maritime infrastructure. This is Sugar Daddy Ward Burns' boat. And then we gravity feed the trout into one of the pens. And you can see how close it is to the shore. That's Newcastle right there. And so this happens in late October. The fish stay in the pen, that pen right there, from October until May when we start harvesting. During that time, the crew is feeding every single day, even in the winter. Even in the winter. Isn't that a beautiful shot? Man, I never get tired of that. So what does this look like from October to May, to June, to July, to August, to September? What does that look like? I'm gonna show you a little bit of data here. And so we get the fish when they're about 300 grams. This is the hatchery growth rate. This is when we're transitioning them into the saltwater pens off Newcastle. And what we try to do is time the water temperatures so when they're in the freshwater raceway, it's about the same as it is in the saltwater system. These transition nicely, and you can see the growth rate increase. They actually doubled in size in about a month. And then comes January or December, January and February, and it gets really cold. It can get down to about 3 to 4 degrees Celsius. They're still growing, but not as fast. Come March, April, you can see the increase of growth rate. And so we're going from 0.6 pounds to 1.5 pounds to almost 2 pounds. And then at the beginning of April, it's about 2.5 pounds. Come May, they're like 3.5 pounds. And this is when we start harvesting. So now we transition from the sampling of growth rates to the harvest. And so these sizes on this side are samples taken from each of the harvests. I'm gonna look at Mike Doherty because it's all his data. I think I got this right. So far, so good. All right, so we start harvesting in May when their fish are about 3.5 pounds. They continue to grow. It seemed like it leveled off at about 5.8 pounds in July through August. These are the mean weights. The maximum weights are a little bit higher. It's a normal distribution of sizes. And so up there, that last point, we're at almost 5.5 kilograms. Multiply that by 2.2 to get pounds. Where's that? 11-ish. The point is, what does this look like? What does this look like? Plots, what do the fish look like? All right, 3.5 pounds, this is about in April, that's the size of the steelhead trout. In May, this is the size of a 5.2 pound fish. This is a rough average of what we're harvesting at, but the biggest ones are over 10 pounds. And this is from October to July. Isn't that gorgeous? Oh my goodness, that's a gorgeous fish. All right, do you know what we do with the fish? You want to make a guess? Broil it? I mean, we have a lot of fish. Bake. Broil, bake, what else?
Smoked.
Smoked.
Yep.
What else?
Sell.
What?
Sell.
Sell them. We sell them. And this is great. This is great because if we're good, not me, but the team is good, and they are, we get income. And it comes income into our program for which we can perpetuate this in the following years. This is exactly what our plan is. Because you know what it's like writing grant proposals? It's like giving a gallon of blood every single time you're doing it. Am I wrong? No. It's hard work. So we sell it. We sell it to our biggest, I can't say our biggest, but one of our most likable partners is Row 34 in Portsmouth. So we deliver, I don't know, what, 200 to 300 pounds a week to them during the summertime, and it's on the menu. It's on the menu Friday, Saturday, and Sunday, and several different ways, from broiled to fish collar, which is amazing actually, to sashimi, to cold smoked, to hot smoked. It's on the menu. You getting hungry? What's that?
We give you a shout out on the credit.
Do we get a shout out on the menu? For credit? At row 34?
I'm not sure.
You're not sure?
They say it's local.
Um,
I think the wait staff talked about
the
it.
wait staff talk about. We actually have taken row 34 chefs out at the site so they can see what we're doing. We also sell to Sanders Fish Market in Seaport, and this is the New Hampshire local fish in the ice case. When we're harvesting more on the order of 1,000 pounds, the market at the moment in Portsmouth can't handle that much fresh product, so we get it smoked. And this is hot smoked. We do that at Boston Smoked Fish Company, and it gets packaged, and we have our own little labels. And at this point, it's not a fresh product, it's a smoked product. It lasts for about 30 days refrigerated or up to a year frozen. So at this point, we're looking into other markets for which we can distribute this. And it's a high value product, actually. And anybody know what this is right here? Roe, is that what you said? That's exactly what it is. Most of our fish are females, and when you gut the fish, you can see the fish eggs in the fish. We sell the roe. This is like a 3-pound batch, and there was like a 10-pound batch 2 weeks ago, and it sells. I mean, we can talk about use of all the parts of the fish, some other talk, but we really like to utilize all parts of the fish. So that was a eye-opening thing for me. And up to date, we are invoiced about $40,000. We're expected to probably exceed another $20,000, or it depends on how well we sell these in the next coming months. It could be even more. We'd like to exceed $100,000 in sales. And at that point, now we're starting to be a sustainable operation. If we can get our costs down. And so we call this sustainable. Sustainable. What does that mean? We have to define that term. We have to define what sustainability is. And we're in the process of doing that. And over the next couple of slides, I'm going to talk a little bit to what we think sustainable is, or responsible might be even a better word. But our fish gets grown here. Our primary market is there. It travels 2.5 miles. There's no refrigerated truck. It gets put on ice. We get the orders, we put it in our truck, we drive it 2.5 miles and deliver it to the clients. If you order wild Alaskan salmon at that same restaurant, it's traveling 3,700 miles. I don't have to say anymore. You can extrapolate to what that means when it comes to what type of human footprint that we have. All right, what is sustainability? And now I'm going to address that a little bit more. And I'm also going to talk more about this multi-trophic thing. What is this multi-trophic thing? And I'm going to do it in the form of numbers, and I'm going to formulate it It's not complicated stuff, but we'll formulate it as if we're engineering a new system, all right? And so we can calculate from the fish how much total ammonia and nitrogen is coming from that fish. And it's a function of the size of the fish or how much fish that we have or what we're harvesting at. And we're feeding that fish during those 8 or 9 months. And as we're feeding that fish during the 8 or 9 months, those fish are constantly putting total ammonia and nitrogen into the water. Total ammonia and nitrogen is a combination of ammonia and ammonium, depending upon the pH of the water. It's mostly ammonium in these waters. And so say we're feeding over that whole time period 10,000 kilograms of feed. There's 0.35 kilograms of protein in each kilogram of feed. The fish, as a biological system, processes that protein into total ammonia and nitrogen. And so then we can calculate how much total ammonia and nitrogen is being output by the fish. If you do your stoichiometric analysis, you like that? Has your chemistry professor ever done stoichiometric analysis? You're not laughing, Larry. You're trying not to laugh, Larry.
I'm evaluating.
You're evaluating. You evaluated me long ago. You can do the stoichiometric analysis and break tan into the amount of nitrogen and then break it out into per fish, and you're producing roughly 43 grams of nitrogen per kilogram of fish. That's what we're putting into the system. If we grow mussels, they are non-fed. The mussels extract stuff. I'm not even quite sure what it is. They extract all kinds of things, from phytoplankton to detritus to whatever they find. If they don't like it, they spit it out as pseudofeces. Did you get that kind of right? Okay, good. But they are made of muscle and shell, of meat and shell. You can do something called proximate analysis. I just learned what this was like a year and a half or so ago, where you can analyze the amount of protein, crude protein in the meat and the shell. You can break that out. There's about 60% nitrogen in the protein. And if you harvest those mussels, you're removing 18 grams of nitrogen per kilogram of mussel in your system. We can do the same thing with sugar kelp. Not as good though. So you can do the same proximate analysis. You can look at how much crude protein is in the sugar kelp. And this is actually our sugar kelp farm right here. You can see the aquafort or the IMGA system in the background. Not as good. 3 grams of nitrogen per kilogram of harvested kelp. So now you want to design a system so you're accommodating or simulating the same amount of nitrogen that's coming from the fish. How much biomass do you need? It's just algebra. And so we can compare it. If we're growing 6,000 kilograms of fish, we're producing 258 kilograms of nitrogen per year. So now, how many mussels do we need? Say we double the amount of mussels. So we harvest 12,000 kilograms of mussels. We can take out 217 kilograms of nitrogen per year. If we do 12,000 kilograms of sugar kelp, we can take out 37 kilograms of nitrogen per year. It's hard to do these things, but then you can just do your mass balance. And these are rough numbers. Michael Chambers has a paper that describes all this. This is not my work. His numbers are very similar, but it could range between twice the amount of mussels to twice the amount of kelp to 3 times the amount of fish. So these are big aquaculture systems that you'd have to have out there. So you need a lot of gear to offset the amount of nitrogen put in by the fish. So this is another definition of sustainability. We have a very, very unique opportunity at this site to throw everything in the kitchen sink from a scientific and engineering standpoint at this. We really want to quantify as many chemical, physical, biological processes that we can. And so that's what we've been doing the last year or so. So we deployed a buoy off the aquaphor. And so this is the aquaphor that we call the IMTA system. It measures temperature, salinity, chlorophyll, dissolved oxygen, dissolved organic matter, turbidity, pH. We're also tracking nitrogen using nitrate, which is actually a part of the nitrification process, if you're interested. We also have an instrumentation package that goes into the system, though we haven't deployed it yet. But most important, and this is the engineering side of it, I like to think, and that we also are measuring currents outside the system, and our plan is to measure currents inside the system. So we're not just getting concentration in and out, but we're getting mass flow rates in and out. If you know the mass flow rate of stuff going in and you've got production or consumption in that system, you can estimate what the mass flow rate is coming out. Knowing the velocities in the cross-sectional area, you can calculate the concentration. Tracking with me on all that? And this is our buoy, and this is the data sets that we get hourly. And you can see that we have an aquadop on here, if you know what that is. And so we're getting velocity profiles external to the system. This is the nitrate concentration from the SUNA instrument, and this is an exosonde environmental sensor that we have on the system. And so what do we like to do? We like to look at the data sets. And so here is a snapshot of representative currents at the site. And so if you look on Google Earth, you can actually see the IMTA aquifer system right there. And we superimpose the results of our data sets, our hydrodynamic data sets. And it's kind of interesting because you have the Piscataqua River coming down like this, and we're actually in this like back eddy formation. So our predominant velocities are actually going in this direction because of a back eddy. At 60 centimeters per second, what I call downstream, 20 centimeters per second, what I call upstream. We can take probably an average value of about 30 centimeters per second. So we can take this information and we can form a simple box model and look at the implications or the interactions of, say, total ammonia nitrogen into the water. And simple mass balance, and so we have flow rate, concentration in, production in the system, and flow and concentration out. We're going to model this as the production of total ammonia nitrogen, which I just went over on a couple slides back. But what we do is you can look at the flow rates and the concentrations, and you can look, and if you assume a certain value, so let's just say the input value is 18 milligrams per liter of total ammonia nitrogen external to the system, because the flows are so strong at 30 centimeters per second through a cross-sectional area of 6 meters by 4.5 meters or so, is such a large flow rate that if we calculate the concentration coming out based on the production of tannin by the fish, it's really negligible. And this is it's a simple box model method to do this, to understand what we're doing here. I mean, we could complicate it by doing computational fluid dynamics or high resolution circulation models or something like that. But this really gives us an idea of the type of impact that we're having. And so this is the first thing I would do if we want to increase biomass, or you want to put the system at a site that has much slower currents. Can we even measure the difference between 18 and 18.0041? Depends on your instruments, your techniques. We don't think we have a very strong impact of nitrogen in the system. All right, so if you're an environmental engineer, that's all environmental engineering. That's all related to water treatment and wastewater treatment, and I really like that stuff. But I'm actually an ocean engineer. I love waves and currents. I like structures, like mooring systems, deploying the gear. It's my bread and butter. So from an engineering perspective, we're also analyzing the system. And so over the last year, we've had instruments, wave spotter buoy, consistently out at the site. We have an upward-looking ADCP that measures directional waves and velocity profiles in addition to the buoy. And we did have an event. We had an event on January 13th. And this is pretty close to shore where our significant wave height, or the HMO, is about 2.2 meters, right? If you assume that the wave height distribution follows a Rayleigh distribution, folks know what I'm talking about here, you can estimate a maximum wave height of, 1.5 to 2 times 2, so, around 4.5 meters or so at this near shore site. The IMK system was there in that storm. And this is Eric Berghahn taking pictures of that structure made of high-density polyethylene pipe and in that storm. And it's sinking, it's twisting and racking. The mooring systems, do you think I slept that night? I did not. You've seen the anchors. Knowing what the anchors, what orientation they are, dug into the sediment, we were concerned. But it survived. We also had 4,000 fish in that system. That was a washing machine of an environment for fish. How many mortalities do we have because of that event? Do you remember, Mike D?
Not many.
Not many. Very robust fish. Long Juan, Zoo, sitting right here, he loves this stuff. Well, I think he does. He builds these numerical models of the system. And so we can take the environmental parameters, we can stick into the numerical model. It's a Morrison equation type model. It's okay, called Orcaflex. And we can simulate the motions, we can look at the net deflections, we can calculate tensions in the mooring line, and we can use it with environmental data sets that we use as input. Numerical models never give you the right answer. So if someone says that they're running a numerical model and it's the right answer, it's not. It gives you a point and a scatter plot of points. And so what we like to do is take some measurements. And so this last summer, we've developed an experimental plan to put load cells on the mooring systems. This is an example of one load cell system. It actually isn't the one that we put on here. It's an older picture. But the whole idea is to measure waves and currents at the same time as the tensions, run the numerical models, and look at the input parameters to the numerical models and develop calibration or validation techniques so you move closer to what the right answer is. Mamoon is a PhD student who is taking this information a step further. He is a mechanical structural engineer. So he's looking at the structure of the high-density polyethylene pipe with these load attachments, with the whole thing twisting and racking. It's a tough problem. And looking at the stresses in the pipe, looking at the cyclical loads on the pipe. Can we come up with a design life to the system? That in turn will work back into the techno-economics of the system, which is directly related to the fish revenues, right? That was site number 1. Sorry, we have 3 more sites left. No, I told you I was going to spend most time on site number 1. But we also have site number 2. And site number 2 is operated by Michael Doherty over here, and it is our educational site. The permit is with the Seacoast Science Center at Odeon State Park. It's really cool because you can wade out to it, and it really engages the public to learn what aquaculture is. We have permits to do eastern oysters, blue mussels, sea scallops, green sea urchins, macroalgae of the brown, red, and green flavors, and the orange-footed sea cucumber. I think that's an echinoderm too. Is that an echinoderm? Yeah, good. I'm not quite sure where the orange-footed sea cucumber hatchery is located. We have a permit for it, right? We also grow kelp. We grow kelp at our Newcastle site, right next to the integrated multi-trophic aquaculture system, but we also grow kelp at our offshore site near the, oh, it's not our offshore site, it's our main site at the Isles of Shoals. And so what we're doing at this site is slightly different with respect to the gear deployed for kelp farming. It's slightly different because in our region, we have an issue. We have an issue associated with the North Atlantic right whale, and it's a horrific issue. There's only about 350 individuals left in the population. They get injured and killed by wrapping themselves in fishing gear, rope, and buoys, and vessel strikes. And what we're trying to do is we're trying to develop rope aquaculture systems that don't include rope. At the beginning of the summer, I would say that our nearshore sites would never have any whales frequenting the waters, but we know that's not the case. These guys have actually seen that whale in amongst lobster pots and the gear that we have out there. So this is a real issue. But using our inshore sites and our kelp farm, we're developing new techniques for rope system type aquaculture, specifically macroalgae or sugar kelp, which the system is typically a long line system. You can grow kelp on the long line system, and it's anchored at 2 points, or you could have arrays of these things, but they're relatively simple. But we're doing it a little bit differently. We're specifying our anchors a little bit differently, and I'll talk about that a little bit later. We are using chain anchors. You can tie a knot with chain, though it's kind of hard. Chain also, it's hard to get caught in the baleen of a whale. It probably could, but there's a thought process for choosing that. But the ropes on the anchor lines, we're not using rope. We're using composite materials, fiberglass rebar type systems. And this is a composite fiberglass, glass fibers, vinyl ester resin. So there's a materials part of this. And the whole idea is that you cannot tie a knot with this. You bend it far enough and it will snap and break, and therefore prevent the entanglement of tightly wrapped ropes around a flipper or a fluke or caught in the baleen. We grow our kelp on high density polyethylene pipe. And it's hard, and this is the actual pipe, and it's hard to tie a knot with high density polyethylene pipe. It does fail, it kinks, but we think that would shed off a whale much easier than tightly wrapped ropes. It's hard to tie a knot. We do have support floats shrouded in PVC pipe. On the floats, we have these things that are used commonly in the pot fishing industry called weak links. And so these are rated at 600 pounds. So if a whale gets caught on that float right there, and it gets caught in the flipper or in the baleen, it breaks at 600 pounds. Provided your anchors are holding, you need to know those numbers. And we pre-tension the system with submerged floats. And so here's my friend and colleague, Michael Chambers, showing how you can bend that fiberglass rod and it breaks. And the whole idea as the whale encounters the rod, it pushes it, it breaks, but you need to have an opposing force. Otherwise, you're just going to drag the gear along with the animal. It's a good pointer, though. It's Friday afternoon. Giving it all I got.
You're not getting sympathy.
He's an awesome guy, that Larry Ward. So not only are we looking at the different gear, but how do we analyze that gear? This is more work by Long Wan Zhu. And so we're developing these numerical modeling techniques such that we can estimate the forcing of the whale on this rod. And it's a conservation of energy technique where we can look at the kinetic energy of the whale in its basic form, 1/2 mv squared. It moves, interacts with the rod, which is potential energy. It's a spring. We can set up the differential equation and we can solve for the force, and then we can model the force distributed over a certain section of the rod associated with the body part of the whale. So if you have a fluke, or not a fluke, but a flipper, it gets caught on the flipper, and the width of the flipper at that spot is here, that much, you can put that into the numerical model. And then what we did, not me, but a graduate student, is we put the rod in a 4-point bending test, and you can break it, and you can determine, this is Louis Gittleman's work, you can determine the bending radius at which it breaks. You can stick this into a model, and here's a model of a whale. And here is the kelp farm in waves and currents. The waves and currents are real here, or they're modeled. And here's the fiberglass rod. And so knowing the force that the whale imposes on the fiberglass rod, we can do that in the numerical routine using that conservation of energy approach. Knowing that the rod will break at a radius of 0.2 meters, you can simulate the breakage of that rod by the whale. That's Long Wan Zhu right there. But the key part of this is that the anchors have got to hold. If you don't have an opposing force, you're not going to force the breakage at that point. It's actually sometimes that's a difficult concept to explain, that opposing force thing. So when we move into the offshore site, now I'm on the final one here, final point. I'll be finished in a few minutes, don't worry. We are concerned about right whales and other large protected mammals at this site. Offshore, 2.5 miles, federal waters are right here. We're about 2.5 miles off. We are permitted to grow and sell blue mussel, scallops, oysters, and all the different flavors of macroalgae at this site. We really are now working on designing a mussel farm for this site, and Jackson Perkins, master's student over here, is doing some of the background work on this now. We were taking the knowledge associated with the kelp farm at the inshore site and working it into an offshore mussel farm. In this case, if we have a backbone of 180 meters with all these mussel droppers, these are the mussel droppers hanging off that backbone. We want, of course, incorporate the composite rods into the system. We're thinking of screw anchors. That may or may not be good. But the idea is we want to be able to produce in a system like this 20,000 kilograms of mussels every 3 years. We don't have gear at the site right now. I mean, we do have gear at the site. We don't have this mussel farm at the site, but we do have gear at the site. And at the site, we are experimenting with growing kelp vertically. The reason why we're doing this is because we're looking at the growth of kelp, or the yield of kelp, as a function of nutrient levels that change with the depth, and the photic zone, which changes as a function of depth. We do have breakaway links on the system, so if a whale hits it, it will break off. But what we're really excited about, if I can get more excited about this stuff, is that we have spat collectors. So we're collecting free-floating baby scallop. Did I say mussel? We have, oh no, spat collectors, scallop spat collectors. So we are collecting free-floating scallop spat in these bags. We want to develop a capability for growing scallops at the offshore site using a system with no vertical ropes. We want to use potentially these scallops in that system. And the idea is to do it all on the bottom at a depth of 35 meters. And how you recover the system is using acoustic release technology. This is being offered to the pot fishing industry. But the whole idea is you don't have any vertical ropes. You grow all your product on the bottom. You do have ropes, but it's sinking rope and it's sitting on the bottom. And we do play with scallops. Here is one scallop in particular, and I think that's your hand, right? So Mike Doherty is also a hand model. And to show you how it works, the whole idea, think about the existing infrastructure associated with the lobster industry. I'll be there. Is that you throw off, this is very similar to pot fishing, except there's no vertical rope. There's the acoustic release mechanism. There's a ground rope. Here's one of the containers that has the shellfish. We've done it with mussels, oysters, and scallops, but we're moving in the direction of scallops these days with our most recent funded projects. And done on a lobster vessel, off the side. And you can have multiple pots in your trawl. The work that we're doing is associated with stocking densities and arrangement of the scallops in these type of bottom condos. So bringing it all together, what do we have going on? We have extensive steelhead trout operation at our Newcastle site. We're moving in the direction of this integrated multi-trophic aquaculture type system, trying to quantify the amount of biomass that we need. We're working with the community at the Seacoast Science Center, whale-safe gear at the Isles of Shoals, hoping to move some of this gear and this whale-safe gear and grow and sell product at our offshore site. And we have a small coastline, 18-ish miles or so, where we honestly feel that this could be a model for other community-based seafood systems. Portland, Boston, we can move to, we're putting a system in the Gulf of Mexico in the next year or so. Island communities, we think that this could be a potential for creating seafood for local communities. And with that, yeah. Thanks. Isn't that cool, that picture? That's the steelhead trout in the IMTA system. These are the bigger trout, and those are the smaller ones that aren't doing as well. Questions? Yes, Larry?
One of the concerns about aquaculture, and it looks like your growth rates are really high and all that, but is the food you're feeding them.
Yep.
So how is that? Are you using a fish species for food, or using fresh water? I mean, grain or?
Well, you are hitting on the most important note in the tune. And he's referring to, these are carnivorous fish. And so in the wild, they eat other fish and other things. And so the feed to be nutritionally productive with high growth rates, the nutrition has got to be right. I would say 25 years ago, I'm not a food nutritionist expert, but I would say 25 years ago, if you looked at the label of the feed, you would probably find fish meal and fish oil toward the top of that label, right? That is the thing that is nutritionally best for the fish. Work has been done to move those two items down to like 6th and 7th on the label. So you know what the number one item is? And I'm going to be transparent on the label. chicken parts. Protein from chicken parts. Protein from the chicken industry. That's not being used. And then the oils are being replaced with plant oils, like canola oil and other type of oils. But you can't there's some things that the fish can't digest, and so you have to be smart with the type of nutrition. But we're moving in the right direction. Great question. Yeah, Larry?
You go ahead.
Yeah, you go.
Nice.
So for your whale breakaway system to
Yep.
farm kelp, say something happens and both anchor points break off, is there a way that you thought of recovering that kelp payload? Because that's what you're farming, so you probably don't want to lose that.
Yeah, you're right, and that could potentially become ghost gear, and that's a big problem. And so what we would do there, if the whole thing breaks free, we would probably have some type of GPS tracking device on the system, where we're actually developing those type of things right now, and some of them are available off the shelf. So if it does float away, you can track it, and you can have either cell phone or radio or sat communications back to shore, so you could go up and find it. Yeah. Yes.
Where can I find this PowerPoint after today, if I wanted to like look back at it?
Right there.
Yeah, I can absolutely give it to you, or there will be a recording of this online on the Vimeo after.
Okay, cool.
John.
Are you quantifying sediment, or is that current?
Oh, yeah, that's a really good question. So what John's referring to is feeding the fish, and there's the other than dissolved parts, nutrients, you have solids, right? You get the poop coming out of the fish. And you also have excess feed. The excess feed is the thing that's problematic because it will sink much quicker than the fecal matter. The fecal matter disperses and it's low concentrations based on the flow rates. So what we do with the excess feed is these guys dive. How often do you dive to check the bottom?
A few times.
A few times. And they do a survey of, are we getting excess feed on the bottom? Because that is indeed an environmental impact. If you're overfeeding and it's just settling on the bottom, you will change the benthic environment. What's that? Lobsters will eat that stuff, I'm sure. Yeah. I'm gonna go with the students, Tom, okay?
Just gonna ask, like, seeing all the fish so close together, what's the risk of like a disease outbreak, or you can't really quarantine one tank from the other? So like, what's the risk to that?
Yeah, there's always a risk. There's always a risk. And so when we're surveying the fish and we're harvesting, and you guys, if I mess this up, just fill in for me. If there is any evidence of, or if we take the mortalities, and there's evidence of something that is suspicious to disease or sea lice, we bring it directly to a veterinarian here on campus and we make that assessment. And we've been lucky not to have that as an issue. Is there anything else I'm missing there?
A lot of importance of this particular site is where it's located. We have great water flow. So good oxygen, good water flow, it keeps the fish healthy and swimming strong. So not by me, we haven't had any major health problems.
Great. Tracy?
Other,
I'll go with you, Tom, next. Go, Tracy.
So I know like the global fishing industry like is one of the sources of plastic pollution, microplastic pollution.
Yeah.
Are there any like advancements people are making in kind of aquaculture, or like I'm just like, thinking about your like HDPE line, like is that gonna degrade and like?
Well, the high density polyethylene is a really stable product. I mean, they're basically replacing, city, towns, water lines with the material. It's a very stable product. The ropes are problematic because the ropes do degrade and they break off pieces of plastic. And Right now, I mean, we could go back 250 years and use cordage that's made out of natural fibers. The core fibers from a coconut, or manila is another one, or there's a bunch of fibers out there. But they were made to be, in old sailing times, they were made to be waterproof by tarring it. And so that leaches, right? And so we don't have a really good alternative right now. I mean, there's dreams of growing kelp and making bioplastics with the kelp, but you know, the margins economically here are very, very slim. And so we're not there yet. However, there is a company in Maine that's making lobster floats out of a fungus. I believe they're mycologists. Did I get that right? Mushroom scientists? I just learned what that word was.
There's an online comment.
Okay, can you read it?
It's from Christine Baran from Melodyne Marine. And she says, an idea to generate revenue, community-supported aquaculture direct to consumers, for example, consumers buy share of UNH. So that's.
All right. Let's have a meeting next week.
Oh, she said you got it.
Tom, you have a question?
I gotta go.
Oh, yeah. All right.
No, catch you later, though. This is all
Okay.
great. Thanks.
Yeah.
What is the long-term goals, like, to market the aquafort for like other industries?
Okay, so that's a multifaceted answer. The near-term goal is to be able to have an efficient operation and system so it can sustain itself, so it can sustain the staff in our operation. From that means we don't have to get grants and so on and so forth to maintain it. Then we can build educational programs around that type of infrastructure that's sustaining itself. And then we'd like to spread that to other parts. And so if when we go to the Gulf of Mexico, we're looking at doing a red drum, a redfish, gracilaria, and oysters on their system. And then figure out the social license part of it down there. Can we engage local industries? And it could be never ending globally. But those are the visions. The vision is on the educational side. Okay. Yes.
How hard is it to get permitted for this if someone wanted to privately go into aquaculture out there without the whole backing of the university and everything?
Very difficult. And so the reason why this is permitted because Michael Chambers right there has a magic wand. No, Michael Chambers and others on the group have built these very strong relationships with transparency, with doing good science, with just a heartfelt approach to it. And so through those personal relationships in the small state of New Hampshire, he was able to get that done. It's difficult. And so we're trying to do the same thing in the Gulf of Mexico. The permitting process is going on for, our second year now? And everybody's involved. I mean, you can imagine the government, state and federal government agencies that are involved to put the system in a small little tiny patch of sand. It's hard.
So is part of the goal to kind of prove that there's not a huge environmental impact and this can be done responsibly and that should be open to private industry?
Absolutely. So we're doing all the science and engineering, we're doing as much of the science and engineering. So if those questions come up, we have hard data to counteract those arguments. But it's also social licensing. This is a small system. It's not a big factory farm. And when people see it, for the most part, they really, really like it. And I think if that spreads on small community scale type levels as opposed to 200 metric tons of product offshore, it's much more palatable. So these are the baby steps. Great question. That is the problem. Billy.
So you mentioned that the eggs are coming from Washington State, correct? And then you also mentioned that you guys are selling the eggs from the females in?
For food.
For food?
Yeah.
Is there any thought of trying to use the eggs that come out of the aquaculture system to then make the next set of fish?
So you're referring to, developing our brood stock?
Yeah.
Yeah. Or using the fish in the pen as the brood stock, but you have to also collect the seminal fluid from the males. Whole another part of the industry. These folks have got it figured out. And so we don't want to try to figure out the egg production side of it. What we are having an issue with is at once they're hatched and in the nursery stage, getting a consistent product. And we'd like to control that a little bit more. Increase the rate for when they get to be able to put into the pens at a lower cost. Control that environment a little bit more, have a little bit more vertical integration. Yeah.
I presume the concentration of nutrients and nitrogen accelerates the growth of mussels. Is that true? And if so, do you have a number?
I don't have a number. And so Skip brings up a really good point. When I'm talking about mass balances, I'm not talking about specific molecules from the fish going into being assimilated in by the mussels or the kelp. That ammonium molecule is long gone. But there's some more that's coming in. And so we're just looking at a regional approach to the mass balance. If you're in a site where the currents are much smaller, or you're putting, and I'm not necessarily mussels, but definitely kelp. This is all naturally occurring kelp along the system because there's a higher concentration of nutrients there. We think. And so can you put the kelp closer in proximity to the fish? I'm not a phycologist. I don't know what the uptake rates are, but I would say that there's evidence to say that you would benefit with the kelp, not so much with the mussels. I mean, you think the mussels are picking up particles directly from the fish?
I believe so.
You believe so?
In
Okay.
addition to the environment, the
Yeah.
phytoplankton, zooplankton that's running through there. And supplements.
Yes.
I have another online question from Aiden Ankerberg. Do you have a list of legislative needs which would help the industry expand in New Hampshire?
Do we have a list of legislative needs to enable this in New Hampshire?
To help it expand.
To help it expand. Do we have legislative needs in Concord?
That's a good question.
Thanks, you too.
Okay. Do you have my contact number? Okay.
Diane Foster is responding, as that's a great question, and she'd be happy to.
Okay. Thanks, Diane.
Follow-up question, you had mentioned that there was improvements that could be made at the nursery up in Ossipee. I
Yeah.
think I've read that statewide, most of our fish nurseries that Fish and Game uses are like 100-some years old.
Yes.
So is part of the problem just that it's an old way of doing things? And we need to kind of see if we can modernize the nursery design?
I can be transparent and answer honestly. I would agree with you 100%. The raceways are technologies that states have been growing trout and bass for anglers, for stock enhancement, for stocking ponds and rivers for 100 years. About that. I'm going to look to Mike Doherty to make sure. 100 years, all right? And it's always been done, not always, but it's predominantly been done over that 100 years in raceways. And what a raceway is you find a really nice brook, and you divert the flow, put concrete walls and grates, and then the flow goes back into the brook again, or the stream or the river. And so from an environmental standpoint, you are going to have large amounts of nutrients going back in. So that's a big problem. And the Merry Meeting hatchery south of the Merry Meeting Lake has that problem. In freshwater, the limiting nutrient is phosphorus. And so they are having algal blooms associated with the phosphorus coming from the hatchery. And so, yes, they really should think about recirculating aquaculture systems, treating their own water, or incorporating hydroponics in the form of aquaponics so you can take those nutrients up and have it in the form of plants that you actually sell. So yeah, it is an archaic way of doing things. It's cheap, but not good for the environment. Great question. Yeah.
I have a policy question to follow on to all of these
Sure.
policy questions. So you kind of talked about the costs you're receiving from kind of exporting your goods to the community, but you also mentioned that you're trying to start this up in different communities. Do you have an idea of what that startup cost would be to develop multiple aquaforts and implement them in local communities, especially ones that may need more government help overseas?
Absolutely. We are the great thing about this team, and I'm gonna look up Mike Doherty again. Michael Chambers has been doing this for 10 years, but he maintains the data. And so every pellet of feed is accommodated. We know where it comes from, we know how much it costs. We have costs associated with fuel, boat rentals, we have labor costs. We had to buy an ice machine this year. We're looking to buy a new freezer because we've got too much product and we don't have a place to put it. We are documenting every single one of these things. And so with that, we're hoping to roll into a techno-economic analysis, and then we can identify where the bottlenecks are and we can put research into those bottlenecks. That is just with this set of products. We would have to do something similar in these other environments. However, we think that we could lessen the learning curve if we do it here using the funds that we have. Yeah. That's totally, that's what we think about all the time, actually. The dollar signs are the most interesting unit in this operation. Yeah.
Was supposed to be a question, do we have like a data on computation regarding the return on investment, how many years before we?
Yeah, so so the question was a computation on return on investment, and we could also talk about internal rates of return. And so we're collecting all the data sets now so we can do those type of calculations specifically for this operation. That is an end goal. Those engineering economics are a big part of what we're trying to do. We don't have the answer. And we are subsidized. I mean, I can't do this for free, you know? We're subsidized.